Hello everyone and welcome to today's live broadcast, Microfluidics Without Borders, Commercialization Perspectives of High Throughput Applications for Acoustic Droplet Ejection Technology, presented by Dr. Sammy Datwani, Senior Director, R&D, Principal Engineer, LabSite Inc. I'm Julie Simmeroff of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. For questions not answered today, we will follow up with participants via email. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the icon located on the lower right of the screen. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. Please join me in welcoming Dr. Datwani. I will now turn the presentation over to him.
Thank you, Julie, for the very kind introduction, and I also thank the organizers of LabRoots for this opportunity. Once again, my name is Sammy Datwani. I'm going to talk about microfluidics and specifically what we do at LabSite. Thought I would take a second to kind of honor someone who's been very influential from a scientific perspective. And really the point I want to drive here is that science is incremental, and what I'm going to share with you is definitely an incremental improvement, but it's got great applications. So to raise new questions, new possibilities, to regard old problems from a new angle requires creative imagination and marks real advance in science. What we're doing at LabSite is essentially turning the world upside down, and I hope you get that message. I also thought I'd take a few seconds to talk about microfluidics and nanotechnology. This is focused on microfluidics and high throughput automation. What is it and why do we care? The Nobel Prize winner, Richard Feynman, in 1959 gave a seminal lecture that is often stated as the starting point for the science of miniaturization and planted the seeds for micro and nanotechnology. Here's a great quote I really enjoy. It would be interesting if in surgery, if you could swallow the surgeon, put the mechanical surgeon inside the blood vessel, and it goes into the heart, looks around, and finds out which valve is faulty, and one takes a little knife and slices it out. So that's the next stage. We're looking at organs on a chip in the next generation of microfluidics. But if you look on this slide here on the left, you have glass chips that were made, and these are part of microfluidic chips that are part of a closed platform. You have a caliper, a sipper chip in the middle, and then you have another chip that was basically enabled for doing high performance liquid chromatography on a chip. And the idea is that you use nanotechnology and you can integrate these components and these applications on a chip and get exceptional functionality. Now, these are all very closed systems. If you would think of an analogy, would be looking at Apple's operating system and how it's pretty closed. What we do at LabSite is we have a very open platform. So I'm going to talk to you about that. So our open platform, if you want to think of it as very enabling, it allows users to do what they want. The analogy I have is kind of similar to what Google is doing. So a quick agenda. I'm going to talk to you about the LabSite technology, what is acoustic droplet ejection technology, a little bit of history, how it works, what can we measure, I'm going to talk about aqueous capabilities. Remember that the world is primarily aqueous, and so what are the key components that we had to solve to enable that? Dynamic measurements, managing meniscus tilt, evaporation, the response to sample variability, and the range of materials we can acoustically transfer. Then I'm going to end with a case study, and that specifically shows you some of the engines we have behind the technology. Next, I'm going to go into some applications. These are things that our customers use, drug discovery for screening, compound management. We have applications in protein crystallography, assay preparation, qPCR and biochemical assay assembly. And then finally, I'm going to talk in depth about a gel electrophoresis microarray. This is essentially a microfluidic platform that leverages our open platform. And then I'm going to end with the acoustic loading of a mass spectrometer. This is basically enabling sort of a high throughput mass spectrometry directly from a microplate well. And I'll end with some conclusions and take questions. So acoustic droplet ejection technology. This is not new. This is kind of goes back to that very first slide. We're taking an example of something that was done historically, early 20s. People were focusing basically not at the fluid surface, and you take a piezoelectric transducer like you see in this technical disclosure at the bottom. And by not focusing on the fluid surface, you had uncontrolled drop size. So you have this piezoelectric transducer, but you're not focusing at the fluid surface. In the early 70s, groups at IBM started focusing at the fluid surface, and you were able to control drop size and trajectory. So that enables things like document printing applications, and folks at Recognition Equipment and Xerox patented a few of these ideas. All the rage now, something called 3D printing, but it really stemmed out of microstereolithography. This is basically parts manufacturer, and this was done in the early 90s. LabSite was founded in the year 2000, and we started with acoustic droplet ejection for life science applications. And in the year 2010, we expanded to aqueous capabilities. So we move liquids with sound. We are physically turning the world upside down. We transfer from a microwell plate into an inverted destination plate. And because of surface tension, small drops have the tendency to stick to surfaces. So we transfer, there's no physical contact. We have perfect sample integrity. We have low energy transfer. We make very consistent drop sizes. They're either 2.5 nanoliters or 25 nanoliters, and we're able to push out about 200 to 500 droplets per second. We transfer to an inverted plate, and because of surface tension and electrostatics, we're able to hold these drops upside down, and this is touchless, and it eliminates nozzles, tips, and improves reliability, precision, eliminates washing, eliminates the need for pipette tips, and eliminates the potential for cross-contamination. We use a technology called dynamic fluid analysis, and essentially we have a lot of signal processing in the background. We're able to automatically adapt to fluid properties and adjust in real time. So we're able to handle a variety of fluids, cell lysates, various reagents, concentrations, glycerol, storage solutions, DNA, RNA. There's no operator intervention, and this miniaturizes processes that were previously not feasible. So we're talking about a 2.5 nanoliter drop, which is about 160 microns in diameter, and that's our discretization. Could you please play the video echo animation?
Sound energy to move ultra-low volume droplets of liquid from a source location to an inverted destination with extremely high precision and accuracy. Echo systems move droplets of 2.5 nanoliters per ejection. To transfer larger volumes of liquid, multiple droplets are ejected from the source at up to 500 times per second. The LabSite Echo 500 series liquid handlers use ADE to transfer liquids from 384 or 1536 well microplates into 96, 384, 1536, and 3456 well destination plates, appropriate for your experimental needs. The ecosystem presents a source plate gripper, and robotically or manually, a filled source plate is placed upon the extended platform. The extended gripper pulls the plate into the ecosystem, where it is deionized to ensure straight flight of the droplets. The destination gripper is extended and inverted to hold a destination plate. The destination plate is flipped and pulled into the ecosystem. Surface tension will keep fluids in the wells of an inverted destination plate. Flowing water couples the transducer to the source plate. The transducer is positioned beneath a well and emits a soft ping of energy. Energy is reflected from each of the three interfaces: the bottom of the plate, the bottom of the well, and the meniscus of the fluid in the well. The ecosystem uses the amplitude of the reflected sound to calculate the level of hydration of DMSO if this solvent is used in the source, and the delay of the final reflection to calculate the depth of the fluid in the well. A well filled to a greater depth requires a longer time for the reflection to return from the surface. The fluid in each well is analyzed, as shown here in slow motion. The information provided by the survey determines that the proper ejection parameters and transducer positioning are used to ensure accurate and precise nanoliter transfers. The actual total analysis time for a 384 well plate is less than 26 seconds. Once the wells have been surveyed, the transducer returns to the first well, focuses the acoustic energy where the survey determined the meniscus to be, and emits a stronger ping of energy. The energy causes a 2.5 nanoliter droplet to eject from the surface and to fly up into the appropriate well of the destination plate above it. Surface tension captures and holds the droplet in the well. Fluid from the same source well can be ejected into another well of the destination plate. More droplets are ejected to increase the volume. Ecosystems can transfer to dry or pre-filled destination wells. A record of empty source wells is kept to help eliminate false negatives. Ecosystems can transfer 5 nanoliters from each well of a 384 well plate to a corresponding destination plate in less than 37 seconds. Cycle time can be less than 95 seconds. When the transfer is complete, the destination plate is extended and inverted to allow another plate to enter the system. Ecosystems can upstack or downstack to plates of higher or lower density. Source plates are easily changed to move from low-density plates into high-density formats. The ecosystems use precise and accurate ADE technology to transfer nanoliter volumes of fluids with no tips or nozzles, saving you hundreds of thousands of dollars annually. Eliminating surfaces that can touch the transferred fluids also prevents cross-contamination and losses to surface absorption, to ensure more sensitive and accurate detection of your best hits. The Eco 500 series liquid handlers are the choice for ultra-low volume liquid transfers.
Great, I'm going to jump to the next slide. So we basically use sonar, Doppler, medical ultrasound to do this analysis in the background, and the user doesn't have to take care of that. We have a variety of instruments that have this capability, this dynamic fluid analysis. They are already out of the box. They're contact-free in terms of transfer. This eliminates tip costs. You can go from any well to any well. You can do cherry-picking applications, direct dilution, arraying applications, plate replication, and you can integrate with a third party in terms of automation. So here's a slide, and I'm going to tell you a little bit about our Access laboratory workstation. It's a benchtop automation solution for the Echo liquid handler. It is targeted, it has modular solutions, and there's a range of device accessories and configurations available. They are designed to scale from sort of assay development to full screening practices. Configuration and options are factory tested for target applications. This comes fully optimized out of the box. They're very quick to integrate. Usually a customer is able to run the system on the first day. You can add new devices and you can reconfigure on demand. We've also developed our own software platform, and that's called Tempo, which incorporates the Echo software applications, and you don't have to write custom scripts or programs. And you can export this information into LIMS and other databases. We've had to get into the consumables business because we need to hear through the plastic. We need to understand how the acoustic energy goes through in order to keep our extreme high precision and accuracy of our performance. So we've gotten into the microplate business. We make plates out of 384 or out of polypropylene or COC materials. And we also make plates in the 384 or 1536 style. These are all SBS or SLAS standard plates, and we even have some microclimb lids, and they're enabling different capabilities to prevent evaporation or hydration if you're talking about materials in the wells. In terms of our technology, we've basically been growing by expanding the fluid range. What we have are plots. On the x-axis is the viscosity, and on the y-axis is the surface tension. And as you move from version 1 to version 2 to version 3, you can see that we're starting to encompass more of this this phase space, this 2D phase space. And currently we are in this third version where we're basically looking at being able to transfer the whole range of surface tension and the whole range of viscosity. One of the artifacts we noticed was protein meniscus influences the behavior in microplates. So you've got proteins that are sticky and they tend to adhere to the walls. Typically someone running a protein in a microwell plate will centrifuge it and they end up having a tilt. Since we use our transducer to both send and receive signal as such, if the surface is flat, you get the signal, it returns. If the surface is tilted, signal doesn't come back. So this is a problem we've had to solve, and it's resolved with some IP that we developed. So we had to invent a way to basically create a solution. The meniscus shape also changes depending on what's in the microwell. So we have 4 different solutions here. You have buffer on the left, buffer with protein next to that. That shows you a slightly tilted meniscus. You have a low concentration of surfactant that gives you a shallow curved meniscus. Let me see if I can highlight that here. And the same thing, if you have a higher concentration, you see a steeper meniscus. What we're able to do with our technology is basically we're able to take a map, a 2D map of that interface to determine how tilted it is, and we use that information when it comes to transferring reproducibly and precisely. Here's another example of how what's inside the microwell has impact. So if you apply the same amount of power, but you have 3 different fluids, you have ethanol on the left, water on the right, and in the middle you have intermediate surface tension fluid, Triton X100. You can see that if you apply the same amount of power, the ethanol drop detaches early, and it also brings with it a potential daughter drop or even a third drop. With the same amount of power, if you apply that to an intermediate concentration of Triton X100, You get one drop and almost a second drop go. If you have the right amount of power, you just get one drop and the mound retracts. So being able to adapt on the fly is one of the capabilities that has allowed us to use our technology in a variety of other applications, which I'll discuss. So what do we get? We get really consistent quality across a variety of fluid classes. Shown here is the transfer volume. So you have both the volume accuracy and volume precision for transferring, and over a variety of fluids. So we're looking at 0 to 60% glycerol, 70 to 100% DMSO, 0 to 100% fetal calf serum, and then 25 to 200% of the CMC of Tridnex 100. So we're trying to transfer 50 nanoliters. So these are 20 drops that are being transferred from our source plate into a destination plate. Our source plate is doped with a solution of sodium fluorescein, and then that's transferred to a destination plate that's upside down. So you transfer 20 drops, and then you bulk fill with some sodium hydroxide, and you quickly run an assay, and you compare the fluorescence to that you would obtain from a standard curve. And here are the results. Basically, you have very precise volume accuracy and excellent volume precision. So since we were talking about microfluidics, I wanted to highlight that this is a very open platform, and we are doing microfluidics. We're just not inside the channel, and I wanted to talk a little bit about that. So the drop really doesn't care. It can go out of a microplate, as you can see here on the left, into a microfluidic device. So you could use it for a loading application. You could also use it for an unloading application from a microfluidic device into a microwell plate. Or you could go from a microfluidic device into an analytical instrument. And think about this application on the right a little bit. This is similar to what we're going to be doing with acoustically loading a mass spectrometer. We can precisely have control over the direction and volume, and you can see we have a stroboscopic image of a drop going through the eye of a needle and into a capillary, so we can easily unload or load microfluidic devices. That shouldn't be a challenge. Here we have droplet ejection into a capillary. There are a lot of capillary-based systems that could be used within microfluidic workflows. So you could be loading your CE that's capillary-based, you could be loading your LC that's capillary-based, and you could be using acoustics to get your sample in. So what's the range of materials and what can we transfer? I mentioned this earlier, 100% to 70% DMSO. We can also do 0 to 30% DMSO. Aqueous solutions, water, salt solutions, buffers, surfactant solutions. I talked about Tridnex 100. We can transfer anionic surfactants like sodium dodecyl sulfate, DNA, primers, probes. We have some customers that use that make siRNA libraries with our technology. We can transfer proteins, BSA, lysozyme, fetal calf serum, all the way up to 100%. Glycerol has been transferred up to 60%. Alcohols, plasma, we've even transferred blood, and protein crystallography kits. We are also starting to look at some of the polymeric solutions, acrylamide, and some others, and also some ionic liquids. So I want to talk to you a little bit about how we do what we do, and that has to do essentially with the signal processing in the background. And if we turn this DFA feature, this dynamic fluid analysis feature, on versus off. So I'll describe the experiment to you. The case study is basically looking at this feature, the software feature turned on or off. And here we have a prototype reservoir plate shown here on the left. And what we've done is we've put the microplate, which is a reservoir, it's a 6-well reservoir. We've put it into our ecosystem. We've taken the skins off. And the reservoir enables you to hold roughly 2.8 milliliters of your sample. And we're able to transfer, this instrument transfers 25 nanoliters at a time, so it's discretizing 2.8 milliliters in 25 nanoliter increments. And we're going to actually put a camera in here and see how that process goes. So we put a side view droplet imaging camera within our instrument, and you can see that highlighted by the circle on the left. Here is our capturing scheme. So we basically have a camera and we have a strobe going, and you're going to see the same droplet in 3 different positions. And here's our triggering scheme. We basically have a delay, and then we start our exposure, and we have three strobes, and then we'll repeat this. So you're going to see some videos, and the videos are going to show you the same drop in 3 different positions. I want you to think about if you've captured this, the physics, then you want the droplet velocity to be the same, which means the space between the drops shouldn't change. So let's jump to those videos. Could you kindly play the first video, MIP off, window 1, please? So I think the message you wanted from that video was the space between the drops. So it's the same drop in 3 different positions, and the space between that was changing over time. And with this dynamic fluid analysis feature turned off, essentially you have a situation where there are some stray drops, you may have noticed that, and the actual velocity is changing over time. So the next video shows you the end of this transfer process. So remember, you're discretizing 2.8 milliliters in 25 nanoliter increments. So it takes a few minutes, so we're just jumping to the video at the end of the process. Could you play the second video, MIP off, window 2, please? I really like that video. It basically shows that with this feature turned off, you have a very chaotic process. The drops are bouncing off each other, they're not getting to the destination, and so this tells me that we don't have something that's sellable. The third case essentially is we're going to redo this experiment, and we're going to turn this dynamic fluid analysis feature on. So this is all the signal processing in the background. And what I want to convince you is that the distance between the drops stays the same. So you see the same drop in 3 different positions. And over the time course, what you will see is that that space between the drops stays the same. And that tells you that the velocity is the same. Could you please play the third video, 6 kV grid on, please? Great. So I think the take home is that the drops, the distance between those drops stayed the same. It's the same drop in 3 different positions. So that also highlights that we have a very dialed in process. We're also moving the destination, because eventually if you have too much fluid that you're transferring upside down, it will fall back down. Next thing I wanted to talk to you about is some of the applications. So with this capability, we're able to do a variety of applications. We're going to start first with arraying. We actually buy a lot of this water-sensitive paper. If you take a source well and you fill it with different surface tension, different fluids with different surface tension, you don't even know what they are. Basically, you can transfer, in this case, we're transferring 100 nanoliters, so 4 drops with that large drop instrument that transfers 25 nanoliters per time. And you see that there are no satellites. And the point here I want to drive home is that if you have a very well dialed in process, you don't have satellites. Applications in drug discovery are echo transfer performance is actually independent of transfer volume. This was published in a drug discovery by John Comley, looking at nanoliter dispensing. It really highlights, so you've got volume on the bottom and you've got the variation in the volume on the y-axis. And if you look at a variety of technologies, as you drive from larger volume down to lower volume, you see that most of the technologies start to have more and more variation, especially when you get down to sort of 10 nanoliters and below, except for the lap side echo. There are problems with traditional liquid handling. Here's a really good example where one of our partners, AstraZeneca, basically discovered a class of cancer therapeutics using the echo liquid handler. These therapeutics would have been missed using the generic TCAN Genesis, which is essentially a pipette-based liquid handler. And what I've actually highlighted here is the results of an IC50 curve, where you've looked at the ratio that's generated by the lab side echo versus the TCAN Genesis. So if the two technologies are doing the same thing, that ratio should be 1. But it turns out that depending on the compound, that ratio changes. So there is something else going on, and we believe that these are some of the problems that get addressed by using acoustic dispensing. Applications in protein crystallography. We had a customer that was interested in transferring fluids from an actual protein crystallography kit. These were essentially the kind of kits, like one example is the Hampton set, and essentially we transferred from a source plate of 30 microliters, and we used a 3 to 4 polypropylene plate. We had a specification set at 94 out of 98 different fluids. So we don't know what these fluids are, we just put them in our microplate and we use the echo instrument to transfer them. The way we did this is we set up a surrogate fluid, and this is glycerol, and we looked at 0 to 60% glycerol concentrations in water, and we did a factory acceptance test to determine the accuracy and precision, and the results are shown here. So very good accuracy and precision. The follow-on is to actually take our technology and do protein crystal formation, and these were results from Seth Harris from Genentech, where he went and did the echo dispense of both the protein solution and then the protein crystallography reagents. And we did this at 25 nanoliters. And you can see on the right that you have these protein crystal clusters being formed. And they are formed in these sort of target wells that have 3 shells in 1 reservoir. We have another customer, this one's from Gilead, that did some more protein crystal formation at the nanoliter scale. I like this because it's just a beautiful image, and it shows you essentially these protein crystals that are formed at the nanoliter scale using our technology. So they show you the same results when you look at the X-ray diffraction. In terms of assay preparation and miniaturization, we've done a variety of work sort of driving to lower volumes. We took a variety of cell and biochemical assay kits, so 4 kits with 11 reagents, and then we use them to transfer and look at essentially the coefficient of variation of the droplet transfer. So we didn't even know what these kits were. We didn't know what the reagents were inside the kits. And the one thing I want to highlight is in the blue circle that shows you that the transfer CV, so that is 1.3% or 0.95%, if you were looking at 2 drops transfer or 20 drops transferred. The next thing to do was to look at a antibody screening with the homogeneous time resolved fluorescence assay. And essentially this is a human cisbio screening assay where we looked at titrating the human antibody from 0.1 to 25 nanograms per well in 2-fold dilutions. We transferred with the instrument the 110 nanoliters of European cryptate, which is the donor, and then we also transferred the acceptor at 100 nanoliters, and then we added 40 microliters of the buffer. The assay was run, and you can see the scheme on the right. Essentially, we had excellent assay results. Our Z prime was 0.83. For qPCR, we also looked at can our instrument be instrumental in sort of driving down volumes, and can it be used for making some of these assays. First of all, we looked at cross-contamination. So we put, this is for a 1536 qPCR test. We put the positive in the top left of the quadrant 1. The positive in quadrant 2 was in the top right. The positive in quadrant 3 was on the bottom left, and the positive in the quadrant 4 was on the bottom right. So essentially we transferred cDNA to the positive wells, and then all the other primers, probes, master mix went to all the wells. And then we looked at the amplification curves and saw what the results were. So here we're doing a qPCR assay. This is a 500 nanoliter assay. cDNA is 500 picograms per reaction. What I want to sort of highlight is that every well that's supposed to be green is green, and those are the positives, and every well that's supposed to be red is supposed to be red. So we didn't see any cross-contamination. We're also able to use the Roche cell lysis. This is the one-step lysis, and look at a cell titration. So we looked at cell densities from 500 all the way up to 10,000 cells per well. This was a 1 microliter total reaction volume, and we looked at the crossing point, which is going to change as a function of the cell density because there's different amounts of DNA. The point I want to drive home is the low coefficient of variation again, so on the order of 1%. We can also use our technology to actually do dilution. So we can dilute cDNA within our instrument to basically avoid having to do hand pipetted steps. And so what we did here is we took the DNA to run a qPCR experiment and did 2-fold dilutions from 25 nanograms all the way down to 1.56 nanograms. And you can see on the right here the actual heat map. So some of the highlights: there's no detectable cross-contamination, it's very reproducible, we generate real-time data, low standard deviations, low CVs, assay can be miniaturized, it's very flexible with our technology, it's one well that can go from any well to any well, it's rapid and very accurate, so you can set up a plate in the order of 12 minutes for a 1536 assay. Applications: I wanted to dive into a gel electrophoresis microarray application. Essentially, can we use our technology for determining and having a high location accuracy of protein solution that's transferred on a gel? So remember, we have protein solution being transferred on a gel, and so you really are fighting diffusion. So the desired features essentially are location accuracy. We want to sort of hit the bullseye every time. There are three examples. We're fully filled if we're hitting the bullseye, partially filled if we're a little bit off, and you could be fully offset. So we looked at that. We looked at the volume uniformity, and this could be determined visually and electrophoretically, and then speed, and how long does it take to perform a full 384 gel well plate transfer. This work was published in Nature Scientific Reports late last year. So here are the results. Essentially, we have quantitative performance of sample delivery using acoustic droplet ejection. Essentially, we get fully filled with ADE, comparing to pin tools, where most of the time you're partially filled, and we actually didn't even bother with the manual pipette. In terms of volume uniformity, the CV and band intensity is on the order of 8%, and you can determine that it's not as good with the pin tools, and then the manual pipette. We did run this, and it was about 29%. In terms of speed, this sort of relates to being able to do a high throughput application. It took the acoustic droplet ejection technology about 3 minutes. Pin tools took about 15 minutes, and the manual pipette about 20 minutes. So once again, I'm talking about transferring proteins or antibodies to a gel, and essentially there's going to be diffusion. And so the faster you do that process, the more accurately and precisely your sample bands will be when you run the electrophoresis. So if you look at the transfers, on the left you have the ADE loading, which takes about 3 minutes, and on the right you have the manual loading. I've blown up 3 wells just below that, and those 3 wells show you essentially the bands that were separated when a potential difference was applied, and then you can calculate the peak intensity. So we determined that using ADE it's 8%, and using manual loading it's 29%. Then we went and worked with some researchers at UCSF. They have these recombinant antibodies, part of the recombinant antibody network, and we were looking at basically looking at electrophoretic mobility shift assays on this gel, polyacrylamide gel, and we wanted to use this technology for the free antigen binding to determine the dissociation constants. So we looked at a concentration gradient, we looked at a variety of different recombinant antibodies, and we determined the dissociation constants. So this also compared favorably with the octet BLI, which is the standard that's used in the industry. So let's look at the results. The advantage of using ADE with this gel over other tools. Essentially, you get more with less. If you look at ADE with this gel, it's about 400 data points per hour. The manual loading gets you a little under 100 data points per hour, and octet BLI, you'll get 1 data point per hour. And that translates directly to the dissociation constant throughput per hour. So the key is you are using much less sample, 3 times 10 to the minus 8 grams with ADE versus 1 times 10 to the minus 5 grams with the octet BLI, or 1 times 10 to the minus 5 with an ELISA. So you're using much less sample consumption in both the antibody and the ligand, and we think that there's a great capability to make this a high throughput application. The next application I'm going to talk about is high throughput mass spectrometry, and we're using our acoustic technology for that. So why use mass spectrometry to screen? Typically people use readers. In a fluorescence-based reader, one would have to add a fluorescence marker. So mass spectrometry for screening is great because this could be label free. It reduces the risk of artifacts. It's lower cost, no complex assay reagents to purchase. You can also do rapid assay development. You can imagine being able to screen the same well many times over, and you're able to get sample kinetics. You're allowed to look at a wide range of endpoints. Could be proteins, lipids, DNA, sugar. You get a true high content readout. So you can look at pathway analysis, metabolomics, and then sensitivities on the order of femtomolar to picomolar. So why hasn't this happened? So this is a slide I'm entitled towards label-free HTS with mass spec. Despite the progress, speed remains the bottleneck. So we're going to look at different techniques and the time per sample and the time to do 1 million samples. And I have a comment associated. LCMS is the gold standard. It takes 1 to 15 minutes per sample, depending on if you're running gradient separations. It will take you 2 years to run through 1 million samples. So it's expensive. You can multiplex to shorten screening time, but you do have solvent and sample consumptions that are different. In 2010, rapid fire was introduced, and this takes about 10 seconds per sample. It would take you then for 1 million samples, about 110 days. So this uses solid phase extraction, and the main application is really in post HTS. MALDI has some matrix issues for small molecules, and they are complex workflows, and you're looking at putting your sample onto a substrate. It takes 1 to 5 seconds per sample, and then for 1 million samples, it will take you on the order of 12 days. What we're calling with our acoustic version of the Echo, we call that the Echo MS. It takes you 0.3 seconds per sample, and you'd be able to do a 1 million compound screen in about 3.5 days. So you can eject and ionize directly from the plate, and we use only nanoliters of sample. We've been very fortunate to work with 2 partners. Basically, Waters provides the mass spectrometer, and AstraZeneca and LabSite together, we're developing this acoustic mass spec platform and trying to put it to good use. So what's the difference? So this is a method that is similar to electrospray ionization, but it's actually a little bit different. With electrospray ionization, you essentially have a sheath gas at high pressure, and you have a capillary, and you are you're basically working in an electric field, and you have a Taylor cone, and then droplets are sort of dispersed from the tip of that Taylor cone. These droplets are charged in the electric field and then make its way into the mass spectrometer. They're desolvated, and the ions turn into signal. With the acoustic mass spec source, we basically are ejecting directly from a microwell plate. We have basically a ultrasonic fluid ejection cone. We dispense drops that then become charged in the electric field, and these are on the order of 1 to 20 microns. So here are the features between ESI and Echo MS. Key ones to point out are sort of sample flow is on the order of 1 to 100 microliters per minute, and for the ESI, and for Echo MS, it's 5 microliters per minute. Droplet size, I mentioned with ESI, it's on the order of less than 5 microns. The Echo MS is on the order of 1 to 20 microns. Similar electric fields, similar inlet temperatures, and similar sensitivity. And I'm going to talk to you about that. So we use our technology to both deliver, and then we ionize samples directly from microplates. And here's a schematic. It's a top view. You see on the right here our microplate. Underneath the microplate is our piezoelectric transducer, and we dispense drops directly into a transfer line. So we have our transducer assembly, we have our microplate, we have the collection nozzle, which applies the electric field, we have a heated transfer interface, and then we have the mass spec on the left. So we're working with the front end, and these are Waters SQD2, TQXS, or the G2S. Here is the Echo MS Alpha breadboard. So we have a few of these installed globally. So we have 4 units in total. 2 are installed and in use for real high throughput screening assays. We have basically a picture. You can see the nozzle, the transfer interface, the mass spec front end, microplate, and the transducer assembly. We've had to come up with a clever way to make sure our microplates don't build up charge. And we've actually patented that, and some of this work is described in the Journal of Lab Automation by Sinclair et al. Here's a photo essentially of the acoustic mist that's created on the right, and on the left is the analogy I talked mostly about dispensing droplets that are on the order of 2.5 nanoliters. Let's take a look at that. So on the left, you have this video where you see 1 droplet coming out, and on the right, you have this burst of this acoustic mist, and that's what is transferred into our interface. Next thing we do, we can sample basically 3 samples per second. So we have a stage that moves, and so we can process 384 well plate quite quickly. We have also enabled automation, so we have fast plate swapping using our robotic integration. So this allows you to have over 8 hours of walk-away operation. Here we're looking at some data acquisition. So this is the extracted ion time trace of an acetylated tripeptide substrate at 601 mass to charge. We are looking at the data from 60 samples processed in 18 seconds. I'm going to talk about this a little bit later. So what you end up with is essentially how do you curate this data, because you're generating so much data. So we have the lab site echo mass spec, and then we have the water's mass spectrometer. They are connected to the Masslynx PC. The information is then sent to gene data for screening. And then the individual wells and are binned. So you have the ions that are binned, and they're basically then processed into individual well mass spectra. So you need to come up with a different methodology, and we have this EchoMS link software that handles and curates data in a timely fashion. Next thing I'm going to talk to you about some preliminary performance. I've shown you how we go from ejection to getting data. First, I'd like to convince you that we have an apples-to-apples comparison with the water's ESI source that shows you that the sensitivity and the detection is very similar. So we looked at the EchoMS versus the electrospray interface performance on the same water's single quad SQD2. We looked at the linear range, the limit of detection, the limit of quantitation, and we looked at two simple analytes, glufib, which is 786 mass to charge in the positive mode, and we looked at warfarin, and that's 309 mass to charge in the positive mode. So here's a graph that shows you the corrected signal versus the concentrations. So we made a variety of serial dilutions and then transferred this using the EchoMS platform, and then also compared that to the acuity interfacing with water single quad. And as you can see that the linearity is very similar, and you can see that the range is also very similar. I'm going to look at the data in a little more. So we're able to use 30 nanoliters on the EchoMS versus 5,000 nanoliters using standard ESI. What I want to highlight is the similar limit of detection and limit of quantitation between the EchoMS for these two analytes, glufib and warfarin. And we actually have a little less background signal. The next thing we did was use the EchoMS for a high throughput compound screen. We looked. We basically have two EchoMS breadboards, one in Alderley Park in the UK and the other in Gatehouse Park in the USA. They are both paired with the water's QTOF mass spectrometers, and they're used for high throughput compound screening. We've completed a 300,000 compound screen for glutathione disulfide reductase, and we have another screen in progress. I'm going to focus on the first screen. So this is the glutathione disulfide reductase, the GSR assay. It's the first screen, and we looked at basically comparing how we perform. So we did an HTS of 300,000 compounds, and we looked for inhibitory or agonist behavior. So essentially you have this dimer that under some enzyme activity converts to a monomer. So the glutathione, the GSH, is also used by cells to prevent oxidative stress and regenerate antioxidants. It eliminates foreign chemical agents, and it can reduce peroxide to water. So it has implications in cancer research. So the GSH oxidizes to glutathione disulfide. This is the GSSG, and then GSR reduces GSSG back to GSH, maintaining the supply. So our output, we are doing a ratiometric measurement. We're doing a ratio of the 611, this is the substrate GSSG, to the product GSH, which is at 306 mass to charge. One thing we've had to do, because we're not using liquid chromatography step, we've had to actually re-engineer the buffers, and this is work that we've done in concert with AstraZeneca. So our workflow is we take 20 microliters of enzyme plus the compound from the screen, you incubate for 10 minutes, you add 20 microliters of GSSG, and then you incubate for 60 minutes, and you quench the reaction, and then you can read this by transferring it from the echo mass spec. Results for determining the conversion, we ended up using the enzyme that one can buy on the shelf, and these were the conditions. We basically have pH buffered with tris at 7.4, 50 micromolar Trinex 100. One of the other things you can do with this technology is you can determine assay parameters in real time. If you wanted to determine the Km for an enzymatic reaction, you could determine that in real time. This would take on the order of 30 minutes. If you were to use standard C, you might be spending on the order of 20 hours. So you have raw signal and conversion as a function of time, and you can do this optimization to determine your Vmax and your Km for your reaction. Over here, we have data from the actual screen itself. So you've basically been able to screen 384 compounds on the order of 3 minutes. You actually have the ion trace here. This is the time trace for the substrate at 611 mass to charge, the product at 306 mass to charge, and then this min, which is essentially the tells you about the inhibitor. So if you take a blow up of the graph on the left and look at just 9 samples, and you can see the inactive, the min, the max, and an inhibitor. The min compound essentially tells you if there's an inhibitor, and in this case you can see that the mass to charge shows you that it is present, and so you have an internal standard. You look at substrate to product ratios, and that tells you information about your actual analysis and whether there is an inhibitor or not. You take this data and you put it into a gene data screener, and then you can do a whole plate analysis, and we actually ran through 240,000 samples that were processed in 66 hours. We ran a concordance test with LCMS, so our technology was able to do this in 4.4 minutes. EchoMS technology would take you about 20 hours. So a small sample was done, not the whole. The result is over 95% cost savings for AstraZeneca. For a 300,000 compound screen, you're only doing that for about $15,000, so there's massive cost savings compared to a labeled screen. There's $50,000 saved in reagents alone. There's a significantly faster than other label-free options. There's a lower false positive rate. There's less than 100 high-quality hits generated, and there's a lower risk of wasting time and money further down the pipeline. The EchoMS also measures proteins, so you can see a charge protein envelope for ubiquitin over here. This is just a mass scan that shows you the different charge states. So with this EchoMS platform, we can directly acoustically ionize, and you can get sub-second sample processing, and this enables a high throughput screening. So our Echo liquid handler is customized with an external stage for 384 well plates and a new transfer line to carry ions to the mass spec. Modified assays can be run without LC cleanup, and that enables real-time assay development, and that's label free. 300,000 compounds are screened, and results were in concordance with HPLC MS. And progress towards a commercial instrument, we've shown you robotic automation, the software to Masslynx, which shows you the echo and the mass spectrometer well sampling. So we have a big data management for curating big data in a timely manner, and we do that via gene data screener. We are looking to expand applications and further commercialize. So quickly, I'm going to give you some conclusions. We have this built-in intelligence that allows excellent accuracy and precision for a variety of fluids with real-time analysis. This is the dynamic fluid analysis I mentioned earlier. It allows you to have real-time monitoring capability for fluids like DMSO that hydrate, glycerol, others. It's robust when you start, it's robust throughout the process, and the system adapts to changing conditions, prevents cross-contamination, there are no tips. There are a variety of applications: drug discovery, process monitoring, qPCR, protein crystallography, biochemical assay assembly, gel electrophoresis, mass spectrometry, and many more. And we're investigating commercialization of new applications for high throughput experimentation. With that, I would like to acknowledge a variety of collaborators, some from UC Berkeley, Professor Amy Hare's lab, Yu-Chen Pan, from UCSF, Carolina Wipsniak, and Michael Hornsby from AstraZeneca, Martin Bachman, Jonathan Wingfield, Ian Sinclair, from Waters Corporation, Steve Pringle, Reese Jones, and then from LabSite, a variety of folks on my team, Eric Sackman, Eric Hall, Luke Gisling, Rich Elson, Richard Stearns, Lars Malhoff, Celeste Glazer, Maria Sontag, Brent Eden, Shri Rajaswari, and a variety of others. With that, I'd like to thank you for your attention and open up the floor for some questions.
Thank you, Dr. Datwani, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up via email after the presentation. Let's get started. The first question is, can you change the size of the droplets?
The transducer that, okay, the quick answer is the transducer that you have has a certain bandwidth and frequency, and that sets the size of the droplet. So you can change the amount that you transfer by transferring 2 drops, 4 drops, 20 drops, whatever you want, but you can't actually change the size of the droplet on the fly. You'd have to use a different transducer.
The next question is, can you use any vendor microplate?
So we've had to get into the business of microplates because we want to ensure the quality and the reproducibility and the accuracy of the transfer. We're not sure if somebody were to make a plate whether we'd have the same sound that goes through that. So we've taken that sort of on us to make sure that in order for us to enable the high quality, high precision, and high accuracy, we make the plates. Most likely we can transfer from any plate, but fundamentally you're not going to get that extreme level of quality.
Next question is, how do you have such precise control of the droplet transfer?
So we make 1 drop at a time, and this technology has been around for a long time. We use a piezoelectric crystal that's embedded in the transducer. We use sound signal that we have absolute control of, and so we're able to do 1 drop very accurately and very precisely, and we repeat that same process over and over.
And we'll wrap up with one last question, and that question is, how many drops can you transfer?
So with the current electronics, we can typically go on the order of 200 to 500 drops per second. So if you take a 25 nanoliter, or if you looked at a 2.5 nanoliter, that works out to be over 1 microliter per second. The 25 nanoliter, you start to get a higher flow rate. But basically, we are looking at ways to go faster and not compromise on the signal fidelity and the drop quality. But typically 500 is the limit.
I would like to once again thank Dr. Datwani for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 25th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.